Okay, right. So, good afternoon, everyone. After a long time, maybe 10 to 12 days, we are again going live with the next chapter of your class 12, that is biotechnology, right? Now, again, it is a very important chapter, keeping in point of view the number of questions. So, almost 5 to 6 questions probably can come from this particular chapter, right? So, it becomes very important to understand the concept of this particular uh, topic. As we have already done molecular basis of inheritance before this, so after doing that topics, this will be easier for you to understand because we'll be dealing with the process of replication. We'll be dealing with the same set of enzymes like restriction enzymes, DNA ligases, right? So it will be easier for you to understand if you have gone through that particular chapter. Okay, right? Now, this is your first page of NCRT, right? So the name is biotechnology, right? So as we know, I can split this term into two different words. Bio has to do with life, or I can say living organisms, right? And technology has to do with the techniques which we use, right? Using various techniques and living organisms, when we get any fruitful product which is for the welfare of the society, those or that technique or that particular field is known as your biotechnology, right? Till your pandemic 2020, that is your corona, which was there, this was not given much hype, but after that we know that corona vaccine was discovered, right? The vaccine was discovered with the help of a technique, that is biotechnology, right? And we were very familiar with the one word that is everybody knew, RT-PCR, right? This was the test which was done in order to diagnose corona, right? This is nothing. RT stands for reverse transcriptase, right? What does RT stands for? Reverse transcriptase, right? Why reverse transcriptase? Because we know that coronavirus was an RNA virus. Means the genetic material which was present in coronavirus, that was RNA instead of DNA, right? So it was repeatedly mutating and creating new variants, right? So it was becoming very difficult to develop a particular vaccine against the strains which were formed during the era of corona, right? So we with the help of technology, that is biotechnology, test was discovered, that was RT-PCR. PCR here means polymerase chain reaction, right? So this polymerase chain reaction we are going to study in detail in this particular topic, that is biotechnology, as we proceed further, right? Okay. Now, if I talk about biotechnology, then there were three definitions given in your first page of NCRT. That's what I've this is not the one. Yes, right. This is the first paragraph of your NCRT. Very important because three different definitions have been explained in this particular paragraph, right? So, initially, now we term biotechnology now we categorize the biotechnology in three different categories, or rather two different categories. One, that is traditional biotechnology, right? Second, that is modern biotechnology, right? Like we all live in both the eras, that is initially our grannies or nannies, they used to be in old era. We used to call it as, at that time was much of traditional time where people were more social, right? There was an nuclear family, was concept of nuclear family was there. Now we have reached into a modern era. So as time passes, even we find changes in the technology. So we have two terms, that is traditional biotechnology and modern biotechnology, right? Traditional technology, when the light was put forward on modern biotechnology, at that time, this word came into existence, that is traditional biotechnology, right? Modern biotechnology was saying that using living organisms, right, we produce certain products which are useful for the welfare of the society, right? Why we are using the word for the welfare of the society, in a short while you'll come to know, right? So when these things were put forward, so it was told that even in initial days, or in traditional era, or in the era when much of the technology was not developed, right, at that time we used to produce curd, which is a very old technology, right? Everybody at home, whether it is a small 6 7 year old girl child or a boy child, will know how do we produce curd, right? We take a curd, in a common language you call it as khatta, right? And in a scientific language you call it as inoculum, right? And you put it in a milk, to be more precise, in hot milk. Hot doesn't mean a boiling milk, right? But whenever we put this curd inoculum in the milk, we ensure that it is warm, lukewarm or warm, or what our grannies or nannies do, they put their finger and they try to touch. If the finger can bear the temperature, that means it is sufficient for for this curd to be inoculated inside the milk. After few hours of incubation, which varies from season, that is your summer to winter, that is 5 to 6 hours of incubation, we get curd, right? A solid, this is your liquid medium, and we get a solid medium of curd, right? So this is nothing but using living organisms, we have prepared a new product, that is curd. Curd contains, here the quantity more was of milk and less was of curd, right? But what we got at the end was curd, and curd shows presence of lactic acid, which gives sourness to the curd, right? So this lactic acid was produced by Lactobacillus acidophilus, right? Lactobacillus acidophilus. These are the bacteria which are present in the curd, right? What they do? They do curdling of milk, right? So the moment they curdle the milk, we get a semi-solid sort of appearance of the milk, and that is what we call it as curd, right? So it was told that this is also a type of biotechnology, because we have used living organisms. Same way the technique was used in case of bread production, in the dough, right? When we put yeast as an desired organism, right? Apart from that, you take flour. That flour can be of various types. Nowadays you get ragi bread, you get millet bread, right? You get multi-grain bread, right? Either of the flour can be used, but what we take in this, yeast as a desired organism. When we add, it results in the production of carbon dioxide, and from the dough, after baking, we get bread, right? So this is a type of biotechnology. So it was stated that biotechnology is into existence since long back, but it was given more of the importance when reformation was done in the field of biotechnology. That reformation gave rise to your modern biotechnology, right? So modern biotechnology is nothing the use of living organisms. To be more precise, we talk about, or we go at a molecular level. During that time, they were not able to isolate a DNA fragment, but now we are able to isolate the DNA fragment. So when we are isolating a particular desired DNA in order to express gene and to produce a particular protein or a product, that is known as your modern biotechnology, right? So now, as we know, definitions are universal. They cannot be changed from place to place, right? So, your European Federation of Biotechnology, that is EFB, it intervened in between, and it said that it put forward a definition of biotechnology which is worldwide accepted, and it is the combination of both modern as well as traditional biotechnology, right? So this was a brief about what basically the biotechnology is. So if I talk about the any of the process, then all these processes, they are when we talk, okay, all these processes, that is biotechnology or bioinformatics, they are specifically based on certain principles, right? There are certain principles which govern a technique. If I am talking about the production of a sound wave, it will follow law of conservation of energy. Any, if I talk about physics, chemistry or biology, if I have any of the technique, it will work on a specific principle. Same way, our biotechnology also works on two basic principles, that is your principle of genetic engineering and principle of bioprocess engineering. You will very frequently use the word engineering here, because we are talking about bio and technology, and we know that most of the techniques or the technologies are used in the field of engineering, right? So here we are doing nothing, but we are using different types of techniques or tools to produce the desired product from living organism. So word engineering is introduced, right? Genetic engineering means we are doing some changes at the genetic level. So the word here is your genetic engineering. Bioprocess engineering, using living organisms, we are processing them in order to produce certain products, right? Using certain, you can say, I use the word bioreactors, right, or tanks. In common language we call it as tanks. It doesn't mean the which are used in the military, but the tanks like your water tank or such structures where you can fill ample of liquid, liters of liquid, right? So they are used in order to produce desired product. So that comes under your bioprocess engineering, right? Okay. Now, if like now we'll be discussing firstly about what is genetic engineering, right? Okay. If I talk about genetic engineering, my main concept behind genetic engineering is to manipulate or alter the genetic makeup, right? In this genetic makeup, either entire, which is a bit difficult, and some part or parts of genetic material, right? Because we'll be frequently using the word fragment of DNA, right? So either what I do, I alter or manipulate the genetic makeup of course, organism, right? We'll be using the word organism, and organism is of course living organism, right? In order, what can I do? Either I can alter the whole genetic makeup, or I take certain part of that particular DNA fragment and I alter that in order to get the desired product, right? That is what we call it as genetic engineering, right? So in the previous class, I had taken, when the offline classes were going on, we had taken few examples, right? We had taken an example of two child. Every day, what problem you might be facing. My child is very intelligent, your child is not, and all. Of course, whose child is intelligent, those parents don't speak, but the other one's parents speak more, that हाँ काश मेरा बच्चा उसके जैसा होता, right? But this is not the fact. I don't advocate this particular policy of a parent that mera bachcha kisi aur jaisa hota. Be happy with what you have. Each child is unique amongst themselves, right? They all have different capabilities to do. Okay, right? Now, just in or not, demotivating any child sentiment and not uplifting the other one's sentiment, I'm just giving an example to explain you, right? Suppose I take two children of my class and name them as child A and child B, right? Suppose child A has characteristics of being sincere. Of course, if he is sincere, then maybe he or she is hardworking, right? And the other one is a bit naughty, right? As he or she is naughty, so easily gets deviated, sincerity reduces, hard work reduces and the result gets affected. But both are mentally capable of doing or achieving the same target, right? But just we are categorizing on the basis of how they appear to us when we see at them, right? Now, both of their parents, they meet and they say that is what example I took, right? They meet and they say that I want to make my child B as child A, right? Now what child B's parent went to the doctor, right, and told that I want to make my child B like child A. So he told that certain characteristics are genetically acquired or certain characteristics are inherited from parents, right? So maybe this characteristic is present in the child's gene, making him sincere or hard working, right? So those characteristics won't be present in child B unless and until I alter the genetic makeup of child B. What I do, I alter the makeup of child B, right? So what was the suggestion given? That okay, I will try to isolate the gene, that is your hard working and the sincere gene of child A, and I will try to put that in child B. So this process is known as biotechnology, right? Or you can call it as genetic engineering, right? Okay, right? So how it will be entered inside? As we are going in a faster zeal, so that will be discussed in the other detailed class, right? So what is my main purpose now? To insert the desired gene or the DNA into the host cell. This is what you call it as genetic engineering, right? This desired DNA will have my desired gene, and when this desired gene is expressed, it will produce my desired product, right? And this will happen in what? This will happen in host cell. At the end of the class, we will study that if I take a single fragment of DNA, right? This is my vector. If I take a single fragment of DNA, right, and I try to transfer it with the help of a vector. In a short while, we'll be discussing what a vector is, right? And put it inside the host cell, right? So as in when the host cell will replicate, it will also, your desired DNA will also replicate, because these are vectors, they are extra chromosomal self replicating material, right? So they are present outside the chromosome, not a part of the host chromosome, and they have a capability of showing self replication, right? As they show the capability of self replication, they have the origin of site of replication, or I see what we had discussed in the process of replication. Here also they have their particular origin of replication, that is site from where the replication will start. Now here I will get only one host cell having the desired DNA. At the end of the class, we'll be amplifying this and we'll be amplifying this host cell, and we'll be producing billions of desired DNA, and ultimately we will get desired product in a large quantity, right? Yes or no? Because when I carry out the process of transforming this with the help of a vector, it will enter into one host cell, right? After that, when one will replicate into two will replicate into 4, right? But this will also take time. So I want this number to be produced in large quantity. I am talking about tons of, you can say, nutrient broth is used where this host cell is inoculated, and billions of this host cells are produced, right? So they will produce desired DNA. Suppose I take an example of insulin. What will I do? The insulin produced by this one small bacteria, it will be in very micro content, right? So that won't be sufficient for me to put it inside the living organism, those who require insulin. Like if I require insulin, it solely depends on my weight. If I gain my weight, my insulin dose becomes. Why do we get diabetic? We get diabetic because upon gaining weight, the insulin which is produced in our body is not sufficient to cut or to act on the glucose molecules, right? Because our surface area has increased, so that cells are not capable to cope up with that number, right? So thus the insulin produce is not sufficient, so glucose level increases in our body and we become diabetic, right? So we require more amount of insulin, so insulin is injected into the body of a diabetic person, right? So the same way, what will I do? The insulin which is produced from one host cell, it will be in very micro content, so I need to produce it in a large amount. So then this particular DNA fragment of DNA, when we will come to know that this is producing insulin, we will amplify that and make billions of copy, and from that we will extract or purify the drug in large quantity, which will be marketed after clinical trials, after clearing the ethical committee, and that is what we take it in the form of a drug, right? So this is your basic concept of biotechnology, what is taking place in biotechnology, right? Okay, now let us stick to a bit of your NCRT, right? It says genetic engineering is, it has written that it is a technique. Genetic engineering is a technique, right, which is used, or technique used to alter the chemistry of genetic material. Either it can be DNA or it can be RNA, but most of the cases it is DNA, right? And we know that from DNA, what is produced? From DNA, we get RNA. From RNA, we get proteins, right? And we know that enzymes are, as I am talking about biotechnology, I am talking about various enzymes, right? So enzymes are proteinaceous by nature, so we get different types of enzymes, right? So if I talk about the second principle, that is your bioprocess engineering, right? Now what is bioprocess engineering? Now I have produced the recombinant cells, right? If I such cells which are produced by the process of biotechnology, they are known as what? They are known as recombinant cells, right? And these recombinant cells now are provided with sterile environment, right? Sterile means free of microbes, right? Not the one which is desired one, but the free of other microbes, right? Okay, after that, what we will do after providing them sterile environment, we are going to produce copies in multiple number, right? So when we produce the copies in multiple number, we get the desired product in large quantity, right? In large quantity. So this is what you call it as bioprocess engineering, right? Yes or no? Are you understanding? Okay, right? Now, whenever I discuss about genetic engineering or this bioprocess engineering, a term comes into our mind, that is gene cloning, right? Now, clone means making copies, right? And gene means your gene or a genetic material. So process of making copies of a particular gene is known as gene cloning, right? If I say a particular gene, means I will only make copies of desired gene, right? I will not make the copies of extra whatever the DNA is 2.2 meter long. I don't want everything to be copied, but I'll be taking the desired fragment of DNA, right? And I'll be making multiple copies of that particular DNA. That is what we call it as gene cloning, right? So you would have studied in your lower classes that suppose I have a cross is done between a cow and a bull. That is what the example I had discussed. Cow, one I will prefer who will produce more quantity of milk, right? And the bull which is there, it will be resistant to disease resistant, right? And it is more of strength or strong. It is strong, right? So when a cross is done between these two, right, we usually do cross between these two by the process of sexual reproduction, right? The generation what I will get, or if that is a cow, your F1 generation, that is filial 1 generation, this cow will have more of milk producing capacity, means it will produce milk in larger quantity, and it will be resistant to disease resistant, right? But it was observed that apart from this, the cow which I took for this particular process, or the female which was taken, was not having any marks on her body, right? Means it was a single color non-patched cow, right? And whereas bull was having certain patches on its body, right? So the cow, ideally what should have happened, that cow should have gained more of milk producing capacity and disease resistant, and of course in some cases strong also, then this will become an ideal cow, right? But nature doesn't allow this. Nature also has its own restriction, right? So the cow which we got at the end of the cross was having more of milk producing capacity, and it was resistant to disease. But as it was a cow, it was expected that there would be present no marks on the body surface. But here F1 generation showed the presence of marks on the body surface, right? So it was what was concluded that what was concluded from this experiment, along with desired characters, some other characters also got transferred. Yes or no? Right? So if this was a natural process, that was which was taking place in nature, that is a sexual reproduction, mating was done between cow and a bull, right, where we want milk in larger quantity. But when I am dealing with small microorganisms, right, and I'm using lot of effort to isolate DNA, from that to identify my desired DNA, yes or no, from 2.2 long meter DNA, I'm taking an effort to identify a small fragment of DNA. From that small fragment of DNA, I need to first purify, then I need to amplify, right? And then I get my desired product, right? After doing such a clumsy process, or a time taking, energy taking, money utilizing process, if I get some other undesired characters transferred, then this will be a fruitless, or this will be not a fruitful process, right? So to avoid this, what we do, we do isolation of a desired fragment. That is, we only take a fragment of DNA which is of our use. So what we do, we isolate, we cut using different tools of recombinant DNA technology, which we'll be studying in a while, right? And we do this particular process. Okay, any doubts? Okay, right, so we were here. So we have, what is gene cloning? The purpose was shown that example was taken cross between cow and bull, right? Okay. Then the history of gene cloning we'll be studying in a while. The first work was given by Stanley Cohen and Herbert Boyer. Right here we had used a cow and a bull that are known as mammals, right? But here Stanley and Boher, they use microorganisms to explain this particular technique of gene cloning, right? And two sets of organisms were used. One was, what was one? One was Salmonella typhimurium, or you call it as typhi. And the other was Escherichia coli, that is E. coli, right? From this, one will be your donor and the other will be your host cell, right? In which I incorporate the desired DNA is known as host cell, from which I isolate the desired DNA is known as the donor cell, right? Hope I'm clear. In which the incorporation is done of the desired DNA is known as host cell, and from which I'm isolating is known as donor cell, right? So what were the experiments done by these two scientists? First, they took the donor cell, that is, I will be writing S. typhi, that is your Salmonella typhi, right? It will have its own genetic material, right? Okay. Now, apart from this, let us talk about one word before, that is, what do I mean by plasmid? Plasmid is an example of vector. Vector is nothing but a vehicle which will carry the desired DNA, right? Or the gene, right? So what is plasmid? Plasmid is an example of vector, right? And vector are nothing but they are commonly called as carrier molecules or vehicles. What they will do? They will carry desired DNA to the, to be more precise, we'll be writing competent host cell. Am I clear? Right? So what is plasmid? Because we'll be using the word plasmid after this. Okay. Right? Okay. Now, what is the important characteristics of this Salmonella? So we had taken Salmonella typhi as a donor, right? Okay. Now what? It was observed that this Salmonella is resistant to certain antibiotics, right? Or R means it is resistant to antibiotic, right? And this characteristic was present in the plasmid of, or you can say, it is present in the plasmid of Salmonella typhi, right? So suppose this is your desired DNA, right? And this codes for what? Antibiotic resistivity. Now what I will do, I will, so when I draw this one as typhi, right, I'll be drawing with the plasmid in order to avoid confusion. This is my, I'll draw complete plasmid. I will make this with the red, right? Here also it goes with red color. And this is the nucleoid of Salmonella typhi, right? Now what I will do, I will isolate the plasmid, because this plasmid has what? Antibiotic resistant, right? So after isolating the plasmid, I don't want complete plasmid to enter into the host cell. What I want only desired fragment of DNA, right? And here desired fragment of DNA is what? The one which provides antibiotic resistant, right? So what I will do, I will cut my desired DNA, right? And what I got at the end, I got my desired fragment of DNA, right? And this desired fragment of DNA will be added into the host cell, right? Now, this whenever I add into the host cell, this technique can be of two types. We discuss about vector, either the one which is mediated by vector or the one which is not mediated by vector, right? So either I can put this fragment of DNA as it is itself into the host cell, or I will take the help of vector in order to introduce that into the competent host cell. So we were talking about Kohel and Boyer's experiment, right? So in this case, what they did, they took salmonella, the nucleoid of salmonella. They found out that there is a plasmid, right? In this plasmid, they have antibiotic resistant capacity, right? So what they did at the end, they isolated the plasmid. From that, they found out this is my antibiotic resistivity. They cut this particular DNA using restriction enzyme, right? What they use, what a restriction enzyme, in a short while we'll be doing it. After cutting this, they got the plasmid and the desired fragment of DNA. After that, what they did, they took another vector, and they, with the help of the vector, the DNA, the desired DNA was attached to it, right? The same restriction enzyme will be used to cut this particular vector, the one which was used previously, right? After this was inserted into the host cell, that is E. coli, and it was observed that this E. coli had gained antibiotic resistivity, right? So this is what you call it as gene cloning, right? This is what you call it as gene cloning. In this process of transforming the desired DNA into the host cell is known as transformation, right? And before this, I have to make my host cell competent to take foreign DNA. Am I clear? So that it will accept the foreign DNA which is coming inside, right? This is about your gene cloning, right? So when one is transferred, at the end, this will be the appearance of a host cell. This is my plasmid and this is my desired DNA, right? So this E. coli will multiply 1 into 2, 2 into 4, and the desired plasmid will also multiply and they will become resistant to antibiotic, right? So this is about your gene cloning, right? And we have the technology, we call it as recombinant DNA technology. That is the technology which is used to produce recombinant cells. Recombinant cells are the cells which will have the desired DNA segment present in them, right? So what will be the steps of this particular technique? Tell me. Yes. First of all, we are going to identify the desired DNA, right? That what we want. The first step will be identification of course, desired DNA. Now I have identified that this is my desired DNA. What I will do next? I will put it into the host cell. That is introduction into the introduction in the host cell. After introducing in the host cell, what am I supposed to do? Right, I've introduced it. Now I don't want that only one host cell with that particular thing. I want many, right? So I will provide it the sterile environment to grow, right? And at the end, what I will do? I will do gene cloning, right? Multiple copies of this particular gene will be made, right? So these are the basic steps of what we call it as recombinant DNA technology, which we'll be studying in detail, right? Okay. Now, before we move to that, how we are going to carry out all this particular process, right? We name that topic as tools of RDNA, tools of recombinant DNA technology, right? So if I talk about tools of recombinant DNA technology, basically I will have how many tools? I will have, tell me how many tools will be there ideally? There are three tools what you are going to study. First, that are enzymes. Second, that are vectors. That is your host cell, right? Enzymes, in order to cut the desired DNA, to save its own DNA. Vectors, they act as a vehicle or the carrier molecules to take that desired DNA into the host cell, right? Now in this we'll be studying three different types of enzyme. This is one of the important topic, right? One, that is your restriction enzyme. Then we are going to study DNA ligase. We have already done this in molecular basis of inheritance. And then we need at the end one that is polymerase, right? Okay. And in this restriction enzyme, you have two types of enzyme: restriction exonuclease, and we have restriction endonuclease. This categorization is done on the basis of how they cut the DNA. When they are cutting on the sides of the DNA, that is ends of the DNA, the exterior part of the DNA, they are known as exonucleases. And when they are putting cut in the middle part of the DNA or somewhere in the center of the DNA or near to the center of the DNA, they are known as endonucleases, right? And we are going to use endonucleases, restriction endonucleases, for this particular technique, that is your recombinant DNA technology, right? Now, whenever we talk about restriction enzyme, we always take or we always give credit to prokaryotes, right? Who discovered restriction enzyme? It was prokaryotes who discovered restriction enzyme, right? Because these restriction enzymes are the peculiar way of prokaryotes to protect themselves, right? So it was observed that when a prokaryotic cell, suppose this is a prokaryotic cell and suppose this is your E. coli, right, is infected by a bacteriophage, right? This is what? This is your bacteriophage. phage, right? This means bacteria eaters. They eat bacteria. They are a type of not virus, or you can say they are a type of virus, but they are commonly known as bacteria eaters, right? What they do is they insert their genetic material into the host cell, right? As we know, we have heard a very good story, or you might have heard from your grandmother or grandfather, कौवे और कोयल की कहानी, a cuckoo and a crow, right? What the cuckoo will do, the cuckoo will always find the crow's nest. कोयल जो है अपने अंडे रखने के लिए क्या करती है, कौवे का घोंसला ढूंढती है. After that it will throw away the eggs of crow and it will put her eggs in the nest of the crow, and the whole complete roosting will be done by the crow. And when the egg will rupture and the young one comes out, the crow is amazed to know that this is not mine, but it is the child of another bird, right? So the same way, what bacteriophage will do? Bacteriophage is like a cuckoo, right? It will put its DNA into the bacterial cell and it will take up the machinery of the bacterial cell and it will express its DNA. So it will inhibit the expression of E. coli's genetic material and it will start expressing the genetic material of bacteriophage, right? So it was found that when this process is known as transduction, that is your bacteriophage infecting a bacteria, right? So after this, what is observed? This is your E. coli, right? The nucleide of E. coli. This is the nucleoid of bacteriophage. It will take out a pin-like structure and create a pore in between of this cell, right? And from that, the nucleus, this is genetic material of bacteriophage, will be inserted into the host cell, right? And the empty bacteriophage will detach from the bacterial cell. Now, as we know, now E. coli has come to know that certain foreign DNA has entered in my body, right? So, what E. coli will do? E. coli will start to protect itself, right? So, it will start producing, or it was observed that E. coli or any other organism, prokaryote, they start producing certain restriction enzymes, right? And what this restriction enzymes do, they are commonly known as molecular, what are they known as? They are known as molecular scissors, right? So they will cut the foreign DNA which has entered into the cell, or they are known as chemical, they are also known as chemical knives, right? So what they will do, they will try to cut the foreign DNA which is present, which has entered into its cell. Now it may happen that while cutting this DNA, some sequence which are repeating over here, the same sequence will be present over here on its own DNA. So first what it will do, it will methylate its own DNA, right? To make sure that if it is methylated, it is my DNA, you are not supposed to cut it, right? So after using an enzyme, let us not discuss. Okay, right? So it will methylate its own nucleoid and it will, restriction enzyme will cut the foreign DNA, right? So from here, the discovery of restriction endonucleases were made. So every time the credit goes to prokaryotes whenever we talk about restriction enzyme, right? Okay. Now, am I clear? Any doubt? Okay, now when I said that restriction enzyme is going to cut the DNA. Yes, so suppose this is my double stranded DNA with base pairs present on both the sides, right? Joined by hydrogen bond. When this will cut, which bond will be broken? Which bond it will break? Hmm? Hydrogen bond. If hydrogen bond will break, then it will break from between. So two strands will be separated. What I will do? There is no reduction in the length of the DNA. Yes, if restriction enzyme breaks your hydrogen bond, right? If it is breaking your hydrogen bond here, right? Then the length of the DNA doesn't change. I don't want entire DNA. I want a fragment of DNA, right? So it is going to break. It is going to break which bond? The bond which is present over here, the bond which is present between sugar and phosphate. The bond which is present between sugar moiety and phosphate, that is your phosphodiester bond, right? So when the phosphodiester bond is broken, I get various small fragments of DNA, right? So it will never break hydrogen bond, because if I break hydrogen bond in between, my length of the DNA is not altered. I don't want complete DNA. I want a specific DNA. So always it will break the phosphodiester bond, and it will give you. And another important thing, if it will break hydrogen bond, right? If it is breaking this hydrogen bond, now these base pairs, the base pairs which are present over here, they will try to attach again. Yes or no? Because they follow the rule of complementarity. If A and T, if I break once the hydrogen bond, if I don't put anything in between, again A and T will bind together. So whatever the work was done by that enzyme is useless, because there was no outcome. Outcome was 0. So definitely it will not break hydrogen bond, but it will break phosphodiester bond, right? Okay. Now, how it works, working of, if I say, this restriction enzyme, as we know that polymerase and other enzymes we have studied. Suppose this is your double stranded DNA and this is your restriction enzyme, right? This is your restriction enzyme. First of all, the restriction enzyme will scan complete DNA, right? If I talk about transcription, we had a Tata box, or we had a region that is promoter, from where the process of transcription will start. Yes or no? Then if I talk about replication, we had ORIC, that is the origin of replication, site from which the replication will start, right? So the same way, this restriction endonuclease will also not start cutting from anywhere. First of all, it will go and scan the complete DNA to identify the site of recognition, right? So after scanning, first what it will do? First step is it will scan complete DNA. After scanning, what it will do? It will identify, or it will find out, identify the site. Which site? Where it is going to stick, right? Because it is moving, and when it will identify and stick to a particular site, this site is known as recognition site. Recognition site of what? Of restriction endonuclease, right? Because we are talking about restriction endonuclease enzyme, right? After that, what it will do? Now it has found out that here, this is my site. So what it will do? It will start cutting, right? So cutting means here breaking of phosphodiester bond, or you can write sugar phosphate bond, right? Okay, now I said that when it was scanning from here, it identified that this is my site, right? So after identifying, suppose this is my double-stranded DNA, right? This is my restriction endonuclease. What it will do? It will scan the DNA. Now it has identified that this is the place where this is my site. So what it will do? It will come and stick over this. Yes. Now how it will identify that this is the restriction site? There has to be something. Yes or no? Like when we were talking about transcription, there were a particular start sequence, start codon and stop codon. I mean translation. When we were discussing about translation, the process will start with AUG. That is coding for your methionine. Whenever the AUG will come, then and then the translation will start, right? In that also you had a Tata box. Here also you have a specific sequence, and this sequence is known as, very important name, palindromic sequence. The name is arrives from your English word, that is palindrome, right? Now what are this palindromic sequence? Suppose I talk about your English language, we write certain words like pop, right? Hydrogen gas burns with a pop sound. POP, pop, from either of the side I read it, it is read same, right? Likewise, I gave example last time, mom, dad, right? These all are palindromes, right? So from both the sides, whenever I read them, they are read as same, right? So restriction enzyme has a palindromic sequence, or the recognition site has a palindromic sequence, right? What is that sequence? Tell me. G, then A, A, T, T. I've written together. T, 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 C. Right? So here what will I have? Let us put it a bit farther. Here. What will I have here? No, G is there, so C, T, T, A, and G. So either I read from 5 prime, from 5 prime to 3 prime, or from 3 prime to whenever I read it from 5 prime to 3 prime side, I get what? The same sequence, that is GAA TTC or GAA TTC. Such sequences are known as palindromic sequence, right? Very important, keeping in point of view your NEET exam. This is a 6 base pair sequence, 1, 2, 3, 4, 5 and 6, and this is the sequence of restriction endonuclease enzyme, right? Like this, a fact what you have to remember, total in all 900 restriction enzymes were discovered from 235 different strains of, from two different 235 different strains of bacteria. Yes, 230, not 235, 2, 230, right? Apart from this, the first enzyme which was discovered, that was Hind 2, right? The first restriction enzyme, the first restriction enzyme discovered was Hind 2, right? Okay, now whenever I say Hind 2 means I am giving a name to my restriction enzyme, and there are total how many restriction enzymes? There are total 900 restriction enzymes, right? So there was a technique discovered to name the restriction enzyme, right? So we'll be discussing naming of restriction enzymes, right? So whenever the commonly used restriction enzyme is eco R1, right? Okay. From this, E has to do with genus, genus and species. Each organism's scientific name has two parts. First is your genus and second is your species, right? Suppose I talk about human beings, Homo sapiens, we write sapiens twice, right? Homo has to do with your genus and sapiens has to do with your species. Suppose I write ran, the commonly what you know, Pisium sativum, that is your pea plant. Yes, Pisium is your genus, sativum is your species, right? Then Rana tigrina, frog, the scientific name of frog, right? So the first part indicates genus and second part indicates species, right? The same way, if I talk about naming a restriction enzyme, I'll be writing the name of the, I'll be using the name of the prokaryotes from which it was discovered, right? So I will have both the parts, I will have genus as well as species, right? So this was discovered from a prokaryote, that is Escherichia coli. Commonly we call it as E. coli, right? So E indicates genus, that is Escherichia. CO indicates your species. R indicates your strain, and this indicates your order of the isolation, right? That is how it was isolated from the lab, not the order of the discovery but order of isolation. Same way, the second enzyme is given in your textbook, that is Hind 2, HIND and 2, right? This was discovered from Haemophilus influenzae. Influenza causing virus, right? So H is for the genus, that is Haemophilus. IN is for species, that is, D is for strain, and 2 is for 2 is your order of isolation in the lab. Please remember, this is the order of isolation in lab, not the order of discovery or order of isolation, because see, there will be hundreds of labs which are discovering this, right? So it is the order of its isolation, right? Okay, now next topic says how your restriction enzyme works, right? There has to be the working of restriction enzyme. So here we'll be talking about working of restriction enzyme, right? So how we know that there are how many types of restriction enzyme? Restriction enzyme are of how many types? Tell me how many types? One which will cut in the center and the other which will cut in the where? Restriction enzymes in the middle, right? So one is your exonuclease and second is your endonuclease, right? Wrong. Nuclease, right? Okay. Now, what exo will do? Will cut the DNA. Okay. Cut the DNA. But to be more precise, we will write will remove nucleotides, because we are talking at a molecular level, right? Nucleotides from where? From end of DNA, right? And here what it will do, it will remove nucleotides from center of the DNA. So ultimately what it will do, it will break diester bond, right? Suppose this is my DNA, in between when I am cutting it, this will be my restriction endonuclease. And suppose this is my DNA where it is ending and it is cutting somewhere over here, then this is my exonuclease and this is my endonuclease, right? Okay. Okay. Now, what did we discuss? Now we'll be discussing working of endonuclease, right? Now, whenever I cut, why I am cutting DNA? What is the reason behind cutting the DNA? To get my desired DNA, right? And second, when I am cutting my vector, because vector will also be cut with the help of same restriction endonuclease. So when the cut is made on the vector, I need to attach my foreign DNA to vector. Yes or no? When I am using restriction endonuclease to cut DNA, my purpose is to produce the desired DNA, right? But when I am cutting the vector, my purpose is this foreign DNA should get attached to that particular vector. Yes or no? First, like this, we cut, we take this desired DNA and then we clone it with the vector. So one cut is made here and the another cut is made here on the vector. This part of vector is removed and the foreign DNA is inserted, right? Okay. So whenever I have to insert something, we'll be talking about the process in which the vector is involved, not the cutting of desired DNA, right? When I have to insert something, suppose I am taking a vector, and I am trying to insert a foreign DNA. Yes or no? So there should be a way to form a bond between the DNA and the vector. Yes or no? Yes. Vector is also what? Vector is what? What it is? It is an extra chromosomal DNA. Yes or no? Right? Which is not a part of its chromosome. Right? So it will also have what? ATGC. Yes? Adenine, thymine, guanine, cytosine, yes or no, right? And this will also follow the rule of complementarity while binding. A will bind with T, T will bind with A, G will bind with C, and C will bind with G, right? By means of hydrogen bond, right? Now, why I am discussing this? Because when I have to cut the vector, I will cut the vector in such a way that my foreign DNA is able to make a bond with the vector, right? Excuse me. Okay, right? So it will form, it should be able to form the bond between, means bond formation should take place between the vector and the foreign DNA, right? So I have two type of restriction endonucleases, right? The one which will cut the DNA at the end, right? I'm talking about what? I'm talking about now restriction endonuclease, and the one which will cut a bit away from the center, right? It will cut a bit away from the center. Okay, right? Okay, so one which will cut at the end, they will cut in the center. They are known as what? Eco are one example of a restriction enzyme that will cut at the end, right? End or away from the center, and the one which will cut in the center. And the enzyme which will cut in the center is just SMA1, not 2. I always write 2. I don't know why. It is SMA1, right? So what we are going to study? The cutting of eco R1. That is how it is going to cut a particular segment, right? So suppose I have taken what? I have taken now eco R1. What is the peculiar characteristics? It will cut away from center, or it will cut at the end. Both mean the same. To confuse you, the word can be asked, option can come in your exam, right? So name the enzyme and name restriction endonuclease that will cut the desired segment of the DNA away from the center, right? So example will be eco R1, right? Okay. Now suppose I take a sequence that is 5 prime and 3 prime to 5 prime. Okay. Tell me one sequence. Let us take the same one. GATTC. Yes. This was what? This will be CTA. I made some mistake. Okay, no problem. Right? Gee, this is not a palindromic sequence, right? Okay. Okay, now what I will do if I want to make it a palindrome? What I will do, I will insert a A. Then it will become a palindromic sequence. It was a palindrome, right? No. GAATTC. It was. Okay. Right? So this is the my sequence. Now what will come here? Restriction endonuclease eco R1. Right? So it has two options to cut. Either it will cut over here, or it will cut over here, away from the center, right? So when it will cut from here, this much fragment will remain. The one I have drawn with the white will remain and this will be cut off, right? So what I will get, the result I will get as this hole will be there, right? Just to ensure that this is a double stranded structure, G will be here, C, then T, T, A, A. Yes, this is your 5 prime, 3 prime, 3 prime, 5 prime. Now here there are open nucleotides. Yes, which are ready to form bond. Yes, now T can bind to X, whatever you add, Y, Z, E. Yes, yes or no? These are open for binding. These sites are open for the formation of bond, right? So this is a part of our desired DNA. Yes or no? Now suppose I have a complementary sequence on vector. So this will come and bind here. Yes, the complementary sequence on vector will come and bind here and form a bond. In this way, the desired DNA will be inserted into the vector. Am I clear? Yes, such ends are known as what? Another important question. Such ends are known as sticky ends, or they are known as cohesive ends. Means these ends will form, they have capability of forming a bond, right? Such ends are known as cohesive or sticky ends, right? Now suppose if I cut the DNA from the center, right? Exactly from the center. So my second example which I had used, that was of SMA1, right? Which will cut it in the center. Suppose this is my DNA fragment with same G, A, then A, T, T, C. Right? I will get here C, T, T, A, G. This is my 3 prime. This is my 5 prime, this is my 3 prime, this is my 5, and this is my 3. Now, what is the peculiarity of this? It will put a cut in the center. So, when it will cut in the center, I will get fragments like this. Yes, here I have GAA, TTC, right? Opposite to that, AAG, and here CTT. Now there is already a hydrogen bond between the two sequences. So these ends, there is no space for another nucleotide to come and form a bond. Such ends are known as very important questions for your exam. They are known as blunt ends, or they are known as flush ends. Flush it off. They are of no use, right? So, what is commonly used? Your eco R1 is commonly used, right? Okay. Then we'll complete the enzyme. Then the next, now what has happened? Vector is there, foreign DNA is there. Now we need to put a glue, right, to stick this foreign DNA to the vector. So what will come? Which enzyme? DNA ligase, right? It will stick it. After this is going to undergo the process of Replication with the help of an enzyme, DNA polymerase or DNA dependent, as we are polymerizing DNA strands, so DNA polymerase. Am I clear? Right? Okay. Okay, right? Any doubts? No? Okay. Then maybe do vector, or vector will be a bit long for you. It is a very long topic. Tell me, have you understood? Yes or no? Have you understood? If you have understood, then please like the session so that your presence is shown, right? Put some comment if you don't understand, otherwise the session will go on in this way, and maybe I won't come next time for the live session, right? Okay, so that's it for a day. We'll end the class. For the offline students, if they see this, we'll be doing PYQs in the next class, right? And the topic which is left, that is your bioreactors, right? Downstream processes, we'll be doing in the next class. And those who have missed the yesterday's important class, that is your PCR process. Maybe this class online will be carried on further, and in this you can study this again, right? Okay. Thank you so much.